now I could get another line of evidence to see what type of condition we were looking at. Is this anemia, or is this some other defect, just to kind of verify my hunch. So here's our probably most significant contribution that we've made to archaeology at this point. And if you're looking here, what you'll see is this bone, and then inside what people generally consider to be matrix or soil. But because she has this background in histology, she kind of questioned it, and she said, well, I'm going to put that under a microscope and see what it is. Well, it turns out that this is actually bone marrow that was preserved for thousands of years. And archaeologists, if you've done archaeology and you've worked with skeletal remains, you're not supposed to wash them, but sometimes people in the past have washed them. A lot of times that part gets discarded. People assume it's just soil, it's matrix, and they throw it out. They've been throwing out bone marrow, basically. So she prepared these slides, and as a good scientist, I wanted to make sure that this is what we were looking at. I couldn't believe it. You can see there's some blood cells. These little pink structures are erythrocytes. The size of them match up to human erythrocytes. We were very excited. I had another student who worked in a different histology lab do some sample preparations too. This is a frozen section of the bone marrow. The purplish part is the bone marrow material, and the red is stained, and this is representing blood material. So there's actually, we're getting the verification. Okay. So she also looked at the bone itself, and she prepared some slides, and we could find these erythrocytes trapped within the matrix of the trabecular bone, which is very exciting. These blood cells are over 3,000 years old. I think that's pretty cool. But it gets better. Turns out, when we actually prepared the bone marrow slide, well, she did it this time, and she took the bone marrow itself and embedded it in paraffin a little bit differently. This is what we were coming up with. Substantial numbers of erythrocytes and the nucleated cells too. So what does this do? It can help me look for the parasites and the malaria ones and the red blood cells. But now that we have these other cells, and by the way, they're probably preserved by some sort of saponification method during decay, where your fat goes through this process where it turns into glycerol, which is a preservative. So this is what we're thinking. There was an article published in the Journal of Archaeological Science that supports this last year too. So we're doing some lipid analysis on it as well. But you can look at other cells and ask questions about things like leukemia. You can narrow it down. When we see chronic hyperostosis, we might be able to get a good idea of what it really is that we're looking at. Okay, so back to sample 5. Remember, I said, remember that one. This is hemozoan, what it looks like when you're doing light microscopy. And because it's a biocrystal, it refracts the light in a certain way. So you can see it right here as well under a microscope. So we looked at sample number 5 again. And this is what it looks like normally. And then when we turned it on and we did the polarized microscopy, which is what you looked at on the previous slide, we saw that there was some birefringence. Now this is just an artifact of some of the bubbles, but there are some actual erythrocytes that are showing this birefringence. This was a sample that gave us the inconclusive peaks, but I sent my colleague at Johns Hopkins the wrong stuff. I sent him the bone. I didn't send him the marrow. So I'm getting ready to send some marrow back to him to run the tests again. Okay, so where does this take me now? Okay, I think I have this nailed down. I think I have a pretty good idea to tell if someone has malaria in prehistoric and protohistoric populations. Well, what I'd like to do is expand that, and I'm starting this 3rd phase of research where I am actually identifying cultures who live in malarious environments and looking at different aspects of their behavior, such as parenting techniques, the population density, subsistence practices, and those types of and compiling them. In particular, I'm using the human relation area files to start this. There's 149 cultures that I've identified that live in malarious environments. What I'm planning to do with this is create this database that can be integrated with GIS software. And it's important spatially because of the way the disease transmits itself across different populations, but also it can be integrated with other GIS projects that look at the vector and the environment. For example, there's one called MAP that I was talking about earlier today, the Malaria Atlas project. So it could be another layer. And it doesn't necessarily have to be limited to malaria, but that's what I'm focusing on. Once these data sets are out there, and in GIS you can do some different types of spatial analyses, or even just different types of analyses that can let you address your own particular research questions that you might have. Currently, right now, I am collaborating with a researcher at the Henry Ford Hospital. He helped provide some insight with the samples, the histology project that we were working on. And I have some students working in his lab as well, helping out right now. There's an undergrad. There's a pathologist who does lipidomics, and he is studying the lipids in the bone marrow sample from sample number 5 to see exactly what you can tell. One, if they're present, and we weren't too sure, but I suspect that's what's happening in this case. And to look at things like, did the person die from a disease, inflammation that might have been related to a parasite, or from an injury, because you can tell those different things by looking at lipids of all things. Who'd have thought? Also, David Sullivan has been helping me since 2008, working on this project, doing the LDMS, the laser desorption mass spectrometry, and the hemosome detection. I'm getting ready to send samples to him of the bone marrow to test for the hemosome to see if he can get verification that's what I'm actually looking at, because that was the only sample so far that we've seen from a histological standpoint that was showing that birefringence. None of the other ones did, which is pretty interesting. And of course, my former advisor, Robert Tykot, and archaeologist in Sardinia, Alessandro Lusai, is one of them. So I also provided you with some publications that I have in progress. I'm also working with mouse models from the Johns Hopkins Malaria Research Institute. I have about probably 6 mice who died from malaria decomposing right now at the Henry Ford Hospital under different environments, so I can do some experiments with histology and maybe even the LDMS. So that one's coming out. I did some slide preparations with their livers to test 2 different staining methods to detect malaria post-mortemly. And what's really nice about malaria research is that it seems to, in times of funding cuts, it seems to at least maintain, if not increase, their funds to fight this disease. So there's many opportunities to seek funding to do this type of research. The Bill and Melinda Gates Foundation is one, NIH, and there's even some professional development grants that could also serve as a funding source that's not malaria specific too. So I thank you for your attention. And if you have any questions, I'd be more than happy to answer them.
So I'm interested in the problems of identifying malaria in children. Do you know about this study that was done at a Roman villa just south of Rome, and where they found the infants in the...
I think they had frog bones in there too.
Yes, yes. Yeah, the interesting thing was that there was definite evidence of rituals,
right.
Revival of pre-Christian rituals that were outlawed at that time, involving the frogs, the killing of some strangling of the puppies, and then the use of the insertion of medical things like various plants. honeysuckle and stuff. And so I was interested if you found anything more identifiable, because they had real trouble.
Right, I think they had ancient DNA evidence.
Yes,
for
they
the
did find
Malaria
the
parasites.
one.
Robert Solares, I think,
Yeah.
was the person, who did that research. Yes. What we found, and it's kind of frustrating because of the condition of the remains. One, they were very fragmented and commingled, and when they were excavated in paste, which was fortunate for me because they didn't clean bones.
Yeah.
Me, and it has all this good stuff, but the way they were actually separated in the lab wasn't necessarily how I would go about it when I was collecting if I were collecting the data. But one of the juvenile skulls did have evidence of chronic hyperostosis. But my thought was that if they had died from a malaria infection, it might have been falciparum, and it might have been one infection cycle, and it's very deadly, and it could have killed them before any sort of bony response could develop. So that's why I'm hoping that the histology and the other methods will help. One of the issues that might have arisen from my research was the sample size. That's one of the problems I found with it. When I collected the samples for the testing, I did it before I actually had contacted this researcher, David, at Johns Hopkins. So I didn't know anything about the sensitivity of the tests or how many samples I should probably run, how many people would be infected in a population, typically like a percentage, if they did have malaria. So if I used a larger sample size, I might have gotten a different result. Also, I used him because he offered, and it was free, I'll be honest.
Yeah.
And some of the readings that he did in terms of ancient DNA, it sat higher because he's used to working with modern samples. And when I went back and I looked at them, they seemed a little bit more suggestive from a forensic or paleontological standpoint that they may have actually had malaria. And the ones that did come up positive were the two species of Plasmodium that we did expect to see in Sardinia. So there's that. And also, sample number 5, when we did the histology, when you consume certain plants, or even better yet, tetracycline, they use it as a marker when they're doing histology because you can study bone growth based on when you introduce tetracycline, it'll leave this band, and then you keep following it through, and it'll leave another band. It fluoresces, basically. In sample 5, we saw this fluorescing, but I'm not positive if it is from some sort of drug, and in particular, it would be madder, which is used in cases of malaria, or has been from ethno-historic standpoint. But it could have been the stain itself. We're not too sure on that because that can also fluoresce as well.
Just a great presentation, great visual material also. I'm just wondering, you kind of mentioned it on the side about these soft tissues, the bone marrow. Just how unique is this exactly to have these kind of soft tissues for 2,000 years old?
That's a good question because I don't think it's as unique or specific to the site as one would suspect. I also, to kind of get an idea if this is what was occurring, because we did publish on it, and here's one of the points that a reviewer brought up. You know, have you tested samples from other sites? I had my advisor send me a sample from Argentina and one from mainland Italy. The one from mainland Italy was from the Neolithic, and we didn't get really anything too good on that one. The one from Argentina had some things that were very suspect, and it supported what we were finding with the soft tissue. I think it occurs more frequently than we realize.